A 2-year-old male was presented to the emergency department with a febrile illness and lethargy. His mother believes he has a sore ear. We received 3 specimens, an ear swab, blood cultures and a throat swab, and observed these results. From the ear swab, a gram stain yielded no pus and contained a mixture of gram-positive rods and gram-positive cocci. A culture showed mixed growth of normal flora. Blood culture showed Haemophilus influenzae isolated from 1 of 2 bottles after 24 hours. From the throat swab, we cultured a mixed oral flora and were able to isolate Moraxella cataralis. To come to our identification, we conducted the following.
The air swab was gram-stained and cultured on hospital agar and McConkey agar at approximately 37 degrees for 48 hours in air, on subplot agar at 30 degrees for 48 hours in air, and also cultured on chocolate agar at 37 degrees for 48 hours in 5 to 10% CO2. Since the patient's mother believed her son has a sore ear, we select girth media that will help identify significant organisms typically associated with ear infections. Hospital agar was used to detect hemolytic activity of the culture organisms, with Staphylococcus aureus, Streptococci pneumoniae, and beta-hemolytic streptococci being the target organisms. Chocolate agar was used to target Haemophilus influenzae and Moraxella cataralis. Subplot agar was used to detect fungi, and McConkey agar was used to isolate Pseudomonas aeruginosa. Standard biochemical tests were conducted, and no significant isolates were identified. The aerobic and anaerobic blood culture bottles that were collected from the patient were placed into a continuous monitoring blood culture device to be incubated for the standard 5-day period. This incubation period has been proven to sufficiently recover a majority of organisms that may present in blood. However, at 24 hours, bacteria was detected in cultures. Blood was generally sterile environment, so when the machine detected growth in half of the bottles, this raised concerns. We conducted a gram stain on the blood sample and found short gram-negative bacilli cocci present. After the positive blood culture result was confirmed and the gram stain has been conducted, we notified the clinician of our findings. This was because the patient has already displayed symptoms of fever and lethargy, and bacteremia could potentially progress to more severe conditions such as sepsemia and meningitis. We continued our identification process by subculturing our blood samples onto hospital agar, which was incubated for 24 hours at 37 degrees in air, onto chocolate agar, which was incubated for 24 hours at 37 degrees with around 5% CO2, and onto subplot dextro agar, which was incubated for 48 hours at 30 degrees in air. Subplot dextro agar did not have any growth. Chocolate agar displayed large, round, smooth, convex, gray, opaque colonies, and blood agar displayed small, pale gray, moist non-hemolytic colonies. When the colonies were tested, they were catalase and oxidase positive.
Due to these results, we suspected that the organism was Haemophilus influenzae, and so conducted growth factor requirement testing using a Haemophilus ID quad plate. A suspension was created from the chocolate agar isolates and inoculated onto each of the 4 quadrants. After incubating for 24 hours at 37 degrees Celsius, with 5% CO2, we found that the organism only grew on the quadrant containing both hemin and NAD, and so identified it as Haemophilus influenzae. The throat swab was cultured on horseblood agar and McConkey agar at approximately 37 degrees Celsius for 48 hours in air, Subarals agar at 30 degrees for 48 hours in air, and also cultured onto chocolate agar at 37 degrees Celsius for 48 hours in 5 to 10% CO2. From observation of these cultures, a mixed growth of bacterium was identified. Of interest were the pinkish-brown colonies growing on the chocolate agar. which appeared as gram-negative diplococci approximately 0.6 to 1 micrometer in diameter in a gram stain. This may have been Neisseria species such as Neisseria gonorrhea, which is a pathogenic in the throat, or Moraxella catarrhalis, which is part of the normal flora of the upper respiratory tract. To determine the correct organism, a catalase and oxidase test was conducted and a DNase plate was set up. All tests returned back positive, and so we identified this organism as Moraxella catarrhalis. Antibiotic sensitivity testing using the UCAS method was conducted on the identified Haemophilus influenzae and Moraxella catarrhalis. A direct colony suspension was created in saline solution to Macfarlane 0.5 turbidity standard, then inoculated onto MHF agar. A Haemophilus species stamper and a Moraxella species stamper were used to add the antibiotic disc to the agar following inoculation. Antibiotic sensitivities for Haemophilus influenzae included cefotaxime and cefuroxime, and resistance was shown against ampicillin, amoxicillin, clavulanate, and tetracycline. Moraxella catarrhalis was sensitive to amoxicillin, clavulanate, cefotaxime, and cefuroxime, whilst being resistant to ampicillin and tetracycline. A nitrocephin disc was also done for both organisms to indicate beta-lactamase production. Both organisms showed a positive result, indicated by a change in colour from yellow to red following the addition of colonies to the disc from a HbA plate.
Now we had identified two potentially pathogenic organisms, Haemophilus influenzae and Moraxella catarrhalis. However, since the patient was not displaying symptoms of upper respiratory tract infection, such as nasal congestion, cough, and facial pressure, we concluded that the Moraxella catarrhalis isolated from the throat swab was part of the normal flora of the throat, and therefore was not the cause of the patient's symptoms. Thus, we proceeded to investigate the Haemophilus influenzae isolate.
Haemophilus influenzae is a fastidious bacterium which forms part of the normal flora of the human upper respiratory tract. More specifically, it is a thin, short, gram-negative coccobacillus measuring approximately 0.3 to 0.5 micrometers by 0.5 to 1 micrometer. There are 6 different types of Haemophilus influenzae, type A to F, differentiated by their distinct capsular polysaccharides, as well as non-typable strains. The capsular structure enables it to evade many of the immune system's defenses, such as phagocytosis and complement-mediated lysis. Haemophilus influenzae type B and non-typable Haemophilus influenzae are responsible for the majority of Haemophilus-associated human diseases. More specifically, the capsular serotypes of Haemophilus influenzae are generally associated with systemic infections such as bacteremia, meningitis, and septicemia, whilst non-typable strains commonly cause respiratory infections such as sinusitis. Acute otitis media can be caused by both Haemophilus influenzae type B and non-typable strains.
As previously mentioned, there are 6 different serotypes of Haemophilus influenzae, as well as a non-typable Haemophilus influenzae. We used slide agglutination serotyping to determine the specific type that was present in the patient's blood cultures. Our isolate was added to different reagents which contained monoclonal antibodies that would only react to antigens from a particular capsular serotype. When our isolate was added to the type B reagent and agitated slightly, agglutination was observed, confirming the identification of Haemophilus influenzae type B. In certain studies, slide agglutination serotyping has been found to yield incorrect identifications when compared to PCR analysis, the most common error being that non-typable Haemophilus influenzae is misidentified as type B. However, we use standardized reagents and a quality control test to limit the potential for error in our identification. In future cases, other methods of serotyping Haemophilus influenzae that could be used include PCR, counter-immunoelectrophoresis, or immunofluorescence. When we had identified Haemophilus type B as the organism present in the patient's blood cultures, we filled out a New South Wales Health Public Health Laboratory notification and submitted it as an electronic laboratory notification to the Notifiable Conditions Information System. We also called the local public health unit to alert them of the HIV infection.
Therefore, from an analysis of patient samples provided by the clinician, we determined that the patient was suffering from symptomatic bacteremia caused by Haemophilus influenzae type B. Based on the clinical presentation of the patient, a potentially sore ear, fever, and lethargy, one hypothesis may have been that the patient developed a Haemophilus influenzae type B ear infection, which spread into the patient's bloodstream, causing our observation of positive blood cultures. However, to substantiate this hypothesis, a middle ear fluid sample will be required, analyzed, and need to be matched as the same serotype that was present in the patient's blood cultures. Regardless, continual monitoring of the patient would be required to see if he developed further symptoms, which could indicate the spread of Haemophilus influenzae type B and the progression of illness to septicemia and/or bacterial meningitis. Septicemia refers to bacteremia that triggers sepsis, which is an extreme inflammatory response to an infection. Bacterial meningitis occurs when the meninges and subarachnoid space become infected with bacteria, leading to inflammation around the brain. Both septicemia and meningitis can be life-threatening, especially for young children. A recommendation was put forward to the clinician that if he observed any symptoms that could indicate the development of meningitis in the patient, such as neck stiffness, nausea, or light sensitivity, he would provide the laboratory with a CSF sample, as there is risk of meningitis developing from symptomatic bacteremia.